okay, with that, I want to go into some cell stuff, cytology. This is where it gets a little bit more palpable when we move away from the chemistry. At least that's what I think. When we look at cytology, that's the study of cells. And a cell is the smallest independent unit of life. It's the basic unit of life. That's bacteria. That's not viruses. Viruses are not living things, but bacteria are. So bacteria are unicellular. Lots of them are unicellular. We are multicellular. An adult human has 75 trillion cells. That's when the bankers say, we are too smart for you, that's why so much money. And then you realize they're just fooling us. But that's a that level question. So the trillion is, I Googled it because I couldn't figure out what to do. A stack of $100 bills is roughly an inch thick. We stack them together and go from Chicago to New York, we get a trillion. That's a lot of cells that we have in our body. Look at all of them are red blood cells. And then look at that, 100 billion is in the nervous system. That's another place where we have a lot of dense cell structure going on. When we look at the size of cells, we got a micron is a measurement, that's a thousandth of a millimeter. A millimeter is that's a centimeter, that's a millimeter, a thousandth of that is like nothing. That's how we measure that stuff. The largest is in the look, I made an arrow hat. Is in the anterior horn cell in the spinal cord. That's the spinal cord. That's like the stuff that goes from the brain down, and then talks to the body. So you cut that through. When you cut that through, that's what you see. And so there's cells in there that are 135 microns. That's the largest we measured. And an ovum, a human egg, is 120. That's what we can see in the naked eye. Is a human egg. And often a good question to know, not on my test, but some SATs or so, is how big is a red blood cell? So it's 5 to 8 microns. It's tiny. 5 to 8 thousandths of a milli of nothing already. We look at shape, also lots of different shapes. We have, the smallest one is in the cerebellum, and that's in here. That's the cerebellar cortex, not that you can figure. I don't know, that picture wasn't too descriptive, but this is descriptive. That's a human egg. So some of them are round. The ovum is round. Muscle cells are like spindly looking things that they can contract. So the shape dictates the function. And a very, very fundamental concept again in anatomy and physiology. The shape, how something looks, dictates what it's going to do. So you're not going to have a square and try to make a car out of it, and there's a wheel. It's not going to work. It's that simple. That simple. So when we look at things, we want to use that novel way of looking at it all the time. It's like, that question, the why question in this sort of discussion is really important. It's not the 5-year-old why because you want a different outcome. It's the why. How does it work that way? Why is it that way? And that brings us to the understanding of things. So cells, they start originally, mommy and daddy, and boom, we got one coming out. But then how are you going to get from that one to like a red blood cells, a nerve cell, and muscle? How are we going to do that? So that's where we have those stem cells, and then they differentiate, they specialize into many different types of cells. Whatever we need. But the fundamentals are from the stem cells. The stem cell, the mom and dad, DNA and all that is where it comes from. That gives us all the stuff we need to make all of these things. So that goes back to the genes. That's why it's so big. So many genes. So when we look at the basic cell structures we have, contain a fluid that is made out of a saline solution. That's aqueous means watery solution. The saline means it's salty. It's like seawater more or less. And then we have proteins in it. And we call that together, that water broth with protein, we call that the xylosol. And then inside the cell we have a round structure generally, that's a nucleus. And around the cell, we got this, we just talked about this phospholipid bilayer. There's another picture of it. That's the cell membrane. Oh. And then we got these things. And these things are little organs. They call them organelles. And they are structures inside the body, inside the cell, that do all kind of different things. So like one makes the energy. That's that mitochondria. One has to figure out how to make protein. One will be helping with separating the cells. One is the nucleus, where we're going to hold the genetic material and all so forth. So let's go through some of them. Oh, well, not yet. Also inside the cell, before we go to the organelles, this is actually a mitochondria right here. But around all of them and through the whole cell, we have a cytoskeleton. So it's like bones inside the cell, for the cell. And so that's a supportive structure. That's kind of a scaffolding type looking thing. You know, it's not like bones that way, but it looks like it holds things together, makes sure the shape is there. It also gives proteins and things to walk, like pathways to walk around in the cell. I mean, that's some really goofy stuff, but it's really cool. So that's a cell, generic cell. So we got the nucleus is a big thing, cell membranes around it, and we got these different organelles. We got this wobbly thing around the nucleus. We got to start working about that, talk about that. Then we got one that's like here looking thing that connects more to the cell membrane, that export things and import things, and so forth. So that's just when you study, you can at some point then see, do I remember what this is? Do I know what this is, and so forth. The cell membrane is the surrounding membrane. That's also known as the plasma membrane or plasma lemma or elementary membrane also. It's again a phospholipid bilayer, and that makes it semi-permeable. That means water stuff can't go through except oil, and oil stuff can't go through. That's half permeable. Inside of it, we have multiple things. We have channel proteins, and a good channel protein, that's right here, that's a channel. That channel can open or close. And if it's open, it lets stuff in and out. If it's closed, obviously it doesn't. So oily stuff can sort of go in and out on their own, but watery type, hydrophilic stuff cannot. And so they need channels to have stuff go in and out. The other thing we have is pores, and pores are smaller channels. Like they let water in and out, for example, as very small molecules, or salt type, sodium, potassiums. Those are more done with pores, not channels. Channels more like for glucose, larger molecules. And then, well, also what we have is we have receptor proteins. And so, for example, we have a hormone. This is a receptor protein. So let's say we have a hormone. Let's say we have insulin that's going to have to tell the cell on the inside to let some glucose in, open the glucose channel. Actually, the insulin is not a good example because that just attaches to the channel, lets it in. But we got other things that cannot penetrate, that are hormones that cannot penetrate the cell membrane because they're watery type. So what they do is they have a receptor on the outside, and then the receptor on the outside feeds into the inside and does some change on the inside in the chemistry. So it's a messenger. So when you do a Bio 20A, talks about a second messenger system. So that's interesting. So some hormones have to act that way. Some hormones are just oily, they just go in and out. They make it easy on them. Oh, this is the picture that I liked in terms of the saturated fats, how these are like all stiffy, and then we have the unsaturated fatty acids, how they make more room because they're kinked. That's that physical structure, how you can see how it makes more room in the cell membrane, how then nutrients, for example, they have a different reaction up here versus here where there's more space. So we can have different responses. So the communication in a cell is the cell membrane. That's probably a very important concept too. It's not the nucleus. It's not like a brain thing. The cell membrane is what communicates. So it's very important that that cell membrane is in good shape. Not too much oxidative stress. So on top of the cell membrane, we got that film, and that film is known as a glycocalyx. So it has a sugary film, sort of a fuzziness on the outside, and that's part of that communication system. One of the most important things about communicating for them is recognizing self from non-self. Is this my cell or is this not my cell? Is this an invader or not? That becomes very important when you talk about the immune system. We'll talk about that in a little later. That gets a little complicated. And then that brings me to the inside of the cell, the intracellular. Intra means inside. Intracellular structures with different functions. So I think of organelles I think of a cell like a village. Then I'm like, what do we need in a village that is maintaining itself? Well, we need an endoplasmic reticulum. You knew that, right? What a name, right? So an endoplasmic reticulum is yeah, you got to get used to these weird names. Is a structure that comes right off of the nucleus, and it's also membrane. It's also phospholipid bilayer. Whatever is membrane-y in these or have phospholipid bilayer around it. And so this structure here is making proteins and lipids. So it's a factory. It's producing things. And then it transports it around inside the cell. Some of this, see, like here outside is like smooth looking, and then it has these little dots on it as we get closer to the nucleus. The dots here are actually little ribosomes, which is another type of organelle. And the ribosome is a protein factory organelle. So this is where we're going to get the DNA thing. Copied, one of those single DNA, not the double thing, the single one comes out and feeds into here. It's like a tape deck. Like the old tapes, it feeds through and the head reads whatever and the music comes out. Well, here it reads these nucleotide, what's the sequence, and then on here it says, well, we need this amino acid and this amino acid and this amino acid put on next to one another. And so essentially then the DNA becomes an amino acid assembly, book that shows which amino acid after which amino acid for which protein. And it has to code that with nucleotides. And so that's going to be that kick. That's going to be that trick, how to do that. So endoplasmic reticulum synthesizes protein and lipids, and it has a rough one. They call it rough, where these little ribosomes are on it. That's where we make the proteins on the ribosomes itself. And then where it's smoother, that's where sort of lipidy structures are made. And then from there, we have the next one that comes off of it. So this is the ER. They call it ER in short, because endoplasmic reticulum is a long name. And afterwards, towards the outside, towards the cell membrane, more superficially, we have another structure known as the Golgi apparatus that's also baggy, but that picks up the created material from the ER and packages it and exports it. So that's the postal service, as far as I'm concerned. Golgi apparatus is the post office. And then it travels inside the cell, and then the other thing it does, it exports stuff. But it's brilliant. Think about it. Like you make a thing, you bag it in a cell membrane, you bring it to the next, from the ER to the Golgi. The Golgi does some stuff and packages and all that, and then it comes out of there, cell membrane around it again. You export it, you fuse the cell membrane with your whole cell membrane, and export it. It's right there. So you reuse that phospholipid bilayer wherever we can use it. It's so versatile. If the body has something they can use all the time, that's great. We don't need to make different pieces of it. Everybody, if you have an assembly line, if you've got the same piece you can use over and over, you don't multiple different ones. You're going to have, at some point, a bottleneck, and you cannot make it all. So Golgi apparatus, endoplasmic reticulum. That brings us to the lysosomes. The lysosomes are another spherical thing, another layery thing, the phospholipid bilayer on the inside here now. we have hydrolytic enzyme mixtures. We have things that can destruct, that can break down material. They can ingest material, they can degrade them. And so that can be as simple as digesting something, but it can be as much as killing something. As a matter of fact, some cells, they perish themselves, they kill themselves, and call that autolysis and self-destruction. They do that with the lysosomes. So if you feel depressed, you just remember there are some cells that perish for you. They're selfless now. Lysosome. So very often, like we take something in, food, the food vacuole, they call that, that fuses with the lysosome, and then we do some stuff with it, whatever we do, but then it can degrade it. Next, we have centrioles, and centrioles are some really interesting, they're really beautiful when you actually look at their symmetry. It's very, very symmetrical. And they help us, they're microtubules, they help us in cell division. What they actually do is, when you look at here, there's one of these on this side, and one of these on this side, these things. You can't see that, but that's what's there. And from there, you got these green rays that come towards the midline. This is the cell right here, the whole cell. And on the midline, these blue things are actually the genetic material. All the genes line up here, because when you make two cells out of one cell, when you make cell division, you grow the cell, then at some point you got to copy the genetic material. And then you got all these organelles and everything, but what's most important is that the genetic material appears in both daughter cells in an equal distribution and everything, because otherwise the daughter cell can't make protein, and that will be a problem. Then it can't survive. And so this is how we organize that. How we're going to, first in a cell division, we copy the genetic material, and then we have these centrioles that make this organized way of lining all of them up in the midline. And then at the end, when it comes to the cell division, each side gets a set pulled towards it, and then we cleave the cell, and we have two cells. We'll have a whole slide on that. You already did that, right? Yeah. All right, good. Centrioles, lysosomes, centrioles, Golgi, and ER so far. And that's the mitochondria. And that's where we make energy. That's the powerhouse. We need energy. So, it's an interesting structure. It looks like a kidney bean type thing, and it has two walls. There's an inner and an outer elementary wall, an elementary membrane again. And the inner one is very folded up. And what happens is, when we get glucose into the system, glucose is broken down into smaller molecules that are charged. And they go to one side of this wall, inside here. And then you have a really, really high concentration on that one side. And at some point you open a pore, and a lot of these molecules flush in, and the force of them flushing in, together with oxygen, makes ATP. So it becomes like, you think of a Niagara Fall or some fall making energy, like the hydro dam or something. It comes like it's that force of these molecules that rush through that then can attach the phosphate onto the ADP and make ATP out of it. But that's for us what we need to know for sure here, because we need to talk about muscles and bones. That's much more important. We need to know the mitochondria is where we make ATP, energy powerhouse. And that brings us to the nucleus. Yes, question. Yeah. The easiest place to get energy from is a carbohydrate. So the glucose there gets cleaved off, and the glucose goes right into that pathway that makes ATP. When we have fats, fatty acids, and amino acids, we can also make energy out of those molecules, but it takes some energy to make them available, like a glucose type available. So that's one reason why the Atkins diet is say, you eating protein, it takes the body energy to make the molecule to then make energy. So the yield is very different. But we can make energy from all those three macromolecules. even though the glucose is using the energy, the fat sort of the storing energy type-ish, and the protein is amino acids are mostly the building blocks for structure, construction material in the body, and so forth. But we can make energy from it. So yeah, the body's very versatile. So good question. Yeah, I didn't mention that. So that brings us to the nucleus, and that's a large sphere on the inside of the cell that houses our genes, and that's the hereditary characteristics. Most cells have a nucleus. Some cells don't have a nucleus, like red blood cells don't have a nucleus. If they don't have a nucleus, that means they cannot divide. Red blood cells don't need to divide. They just need to carry oxygen. That's all they need to do. That's all we worry about then. We got lots of them. So they get made somewhere else, and then when they are done, they just get discarded and get taken apart from the spleen, get disassembled. Some have multiple nuclei, like muscle cells. When you look at a muscle is like from here to here. That's one muscle. That's a long cell. One muscle cell goes the entire length of a muscle. You have many muscle cells. Like a cell is a strand. It's very small, very skinny, but it's long. And so when it's made, it starts as multiple cells that then fuse together, and the nucleus is left behind. So that's why we have more than one nucleus in a muscle cell. Not that important. The most important thing is if we need to have a nucleus to be able to divide. No nucleus, no division. The nucleus is two elementary membranes filled with pores that is a boundary from the cytoplasm. So the nucleus is here, and it's almost like a cell within a cell. It's separated from the rest, so it's protected. And then inside that, we have another one called a nucleus. I know, why do they do that to us, right? It's like a Russian doll, smaller, 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 and one more. But when we think about it, it makes sense, because we need a place where we're going to make the factories that make the protein. Because that's a protein in itself. Who's going to make that one? The first one. Who's going to make the first one that then we can make it? So that we need a place where that's happening. And that's in the nucleus. So they call that the ribosomal RNA. I just read the word ribosome, and then that says protein factory from the slide. Yep. But that's why we have that nucleus, because who's going to start out making protein? And that brings us to this next interesting part, the genes, the chromosomes. Chromosomes are the carriers of our hereditary characteristic. One gene, so we call this gene stuff we talk, that's information to make one protein. So now we have a definition of a gene, so that's good. One gene for one protein. The chromosomes, therefore, are a cookbook of our proteins. We have 46 chromosomes, 2 sets of 22, mom and dad. That makes us a diploid. We have copies of 2. It's good to have copies. One's array, maybe have another one, we can use that one. But if we only have a one setter, they call them a haploid in that world. We have about 30 to 40,000 genes that are packed in a DNA double helix of about 2 meters in length. It's about this much in length. Every cell has this much in length of a strand in it of that. That's a lot. But 30 to 40,000, think about that. So that needs to be very well organized. So the DNA double helix here is coiled around these protein things called histones. They're just coil things. And then it's moved around, and then it's a strand. And that strand is known as chromatin. It's a spaghetti bowl of spaghetti. That's how it is when the cell is just moving around. And then it gets organized into these chromosomes when we have cell division. And when we look at chromosome stuff, we see these. Right? When we read the table or so in college. This is college, right? Yeah. I remember we had some on the wall. But then that's the chromosome, so we need that for mitosis. Mitosis means cell division. The centromere or kinetochore is where the chromosomes come together. right there. When we make a protein, what we basically need to do, we need to unwind everything back to being in a double helix, and then take the double helix and unwind that, and then we expose the nucleotides, because the nucleotides are what's going to code for the protein. So that's then that genetic code that we need to talk about. So we have all these, well, we don't have all these, we have these four, we actually have 5 types of nucleotides, but we're 4 having DNA. So we got the A, T, the G, and the C. And they come like on a strand. Here, see, here's the double helix. We unwind it and we'll take a little piece out of it. So this is like a nucleotide here, blah, blah, blah. We take 3 of them, nucleotides, and that 3 will code for 1 amino acid type. So that's how we can have genetic stuff, the nucleotide strands, the chromosome, and make proteins out of it. So we take 3 of these letters, and we need to make 20 different one of these amino acids. We only have 20. So we're going to make these tables where we can say, okay, a guanine, an adenine, and then a cytosine make aspartic acid. That's an amino acid. You don't need to know these amino acids, but that's what these are. And you don't need to know the different ones. I need you to know that these are amino acids, and amino acids make proteins. And so that's basically making protein right there. So 3 nucleotides that then will code into 1 amino acids, they call that a codon. Or they also call that a triplet, because it's 3 nucleotides into 1 amino acid, so a triplet goes into 1. And that's when you see sometimes, you know, they have the genetic code, G, G, A, B, 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 B. That's what that is. That's exactly what that is. Nucleotide sequences. So when we take protein, when we make protein, basically we already did it now. Proteins want to work in the body. Yeah, we talked about that. Contract the muscle, hemoglobin. But what we need to do is we need to go and unwind the DNA double helix, wherever that gene is that we want to copy, that we want to make a protein out of. We need to make a copy out of that gene. And now, instead of a DNA, the copy is called RNA. So DNA, when you look at the whole name, it means deoxyribonucleic acid. And RNA is not deoxy, it's just ribonucleic acid. So that's the short version of it. So for us, what we need to know is the DNA is that double helix thing in the nucleus that holds all the genes. The RNA actually, I should say mRNA. RNA stands for multiple things. The one that this is the mRNA. Which is, once we talk about the different types of RNA, when we use the word RNA, we mean basically the copy of the DNA. And then that copy, that was a long discussion on that, that copy then leaves the nucleus through one of the pores. Because we have a copy, if it gets destroyed, it's okay. It's a copy. We still have the original inside. The original will not leave the nucleus. And then that copy goes through the ribosomes, and on the ribosomes we read these triplet things, these codons, and this is also called an RNA. And again, I don't know why they have the same word for all these different things, but we have to just explain it once or so. But we have then one RNA that can read the codon on one side, and on the other side it knows which amino acid goes with that read. And so it just, that way the strand goes through it, and we get the ribones come down and they attach one amino acid after one amino acid, and at some point we have a protein. Like a hemoglobin, I think. No, I don't know how large that gene is. I shouldn't say that, but I don't know it. But it's not that long, like the hemoglobin, so it's not that long. So before we do that one more time, with a few more words on it, I have this slide on RNA. And so the one that's the copy of the DNA is known as the messenger RNA, the mRNA. So that's the copy. So these are the nucleotides sticking out, the different ones, the 4 different ones. We also have the ribosome itself is also contains RNA, ribosomal RNA. That's the one that they make that in the nucleus. So they say this is rRNA. I think of that as the ribosome itself, since it's part of the ribosome already. That way I keep them separate. I know which one's what. And then they have another one to just everything comes in threes, right? So they call that the transfer RNA or tRNA. And transfer, that's the one that on one side can read the nucleotide, and on the other side understand the amino acid that is corresponding to it. So that's actually the one that really, in essence, makes the protein then at the end, because it reads the mRNA. Confusing, huh? A lot of words. Yes, question. Good.
It reads the mRNA and then it does what with the acid? It makes.
Yeah, let's do it. You mean the tRNA? Yeah, the tRNA has the ability to, on one side, it has nucleotides that correspond, to the ones that come from the mRNA, from the DNA part, from the nucleus. And so these are always there. They don't go away. These are just readers. And then the other side has the amino acid on it. And a protein is ultimately a chain of amino acids. And so this is the one then that can read where we are in the sequence and can bring the proper amino acid in and then put it onto the chain of the other amino acids. That also happens here. So when we look at that, that'll be this part here. So this is just another visual, has a couple more names in it that I need to put in. This is one of the two main concepts for cell chapters. So if this is a little goofy, we'll do it again on Wednesday. Wait, today's Wednesday, on Monday. So again, the first step is we have that DNA and we need to make a copy of it. So it's a double helix, so we unbind it. Look at that, I even put the enzyme in. See that word here, ASC? It's an enzyme. I don't need to know that enzyme. But next time in study you come by, it's like, oh yeah, there's this enzyme. Oh yeah, it was there already. And so, but you need to know if it's an ASC, you know it's an enzyme. That makes the mRNA. So the DNA gets uncoiled, and we make a copy, and the copy here is the blue strand. That's the mRNA, and then that leaves the nucleus. And so that copying process is known as transcription. You're transcribing. You go to the court and you have somebody sitting in the front of transcriptionist. They're transcribing the spoken word into a written word. Again, it's still the DNA and the mRNA is the same language. It's just a copy. So they call that transcription. Then that goes out of the nucleus, and then from there it goes into the ribosome. And inside the ribosome, we feed these codons in, these nucleotides in, and the tRNAs come in, these transfers. On one side they carry an amino acid. On the other side they're reading all these codons. And that way we're going to make from nucleotides translated into an amino acid chain. So they call that translation. Because that's like you go from Chinese to Spanish, or from Swiss German to English. That's what happens in my head sometimes, or the other way. But it's a different language. It's a totally different language. So they use the word translation for that, describing that process because of that.
So that makes one protein, right?
All that makes protein. So this is exactly the same process. I just added the translation, transcription words to it. That way we do a few different levels on it. But again, we got to keep it as simple as possible, because the body doesn't have these words. We just need to give it these words to make sense of it. So as your essence inside of I'm a physical person, a kinesthetic. I touch people for a living and massage their muscles and crack their necks. And so I need to feel things. And this is, I need to visualize. I feel it to visualize. So I close my eyes and say, okay, The double helix unwinds, we make a copy of it, the copy goes out, it gets fed into this other thing, and it reads it and brings amino acids in. And then I can do the words. The process is more important. The words, so that's why if you don't know the words and you're thinking you're just simply studying, memorizing words, that's when we got to talk. That's why we'll see if these questions are a little helpful over time. All right, good. Any questions to this so far? Yes?
So if the DNA is made up of 2 nucleotides,
Strands.
and the RNA is a copy, and it only has 1 nucleotide?
Oh, good question. Yeah.
Where's the other?
Since you have complementary base pairs, you basically have one strand goes one direction and the other one is the same thing, just going the other direction. It's the same sequence of nucleotides because they're all complementary. So we don't need both strands to read. They're copies. So we only need one copy to make the protein. Hopefully it's the not damaged copy. That'd be nice. But so we don't need both to make the protein. We only need one single one. And then there's actually one more thing, and I'm not sure where it's written down, but if you think somewhere, have you seen the word uracil? Uracil.
The ribonucleic acid.
Yeah, look at this here. The mRNA, instead of thiamine, it uses uracil. One more of those names. So now you got 5 letters. But the thing is this, if you see something like the genetic code, like this, like I think there's even a test question that just like is, which of these is an RNA? And you see a line of letters. If there's a U in it, you always know it's an RNA. DNA does not have the uracil, the U in it. I know, right? A U in it, it's RNA. Well, if you're looking at the DNA RNA thing, yeah. Well, you can also go with, if it's got the thymine in it, the T in it, then you know it's DNA. But the U is just easy to see, and it's very specific. Good. Look, if you're done with that, questions for now. We'll pick it up next week, but we can go to mitosis. You ready for that? Good, because we're starting to fall asleep, huh? Cell division is very important for growth and cell renewal. Not all cells divide. Generally speaking, the more specialized cells are, the less they most likely divide. So that goes for a lot of brain cells. And then some cells don't divide if they don't have a nucleus. Before we divide a cell, which is the mitosis, we have to copy the DNA. And now, same thing. As making a copy for an RNA strand, we do the same thing, but now we do copy both strands. So now we unzip one and then copy both and end up with 2 of the whole strand thing. Not RNA, now it's 2 DNAs. So, but it's a copy question again. And then once we copy that thing, the next question, the real mitosis question is, how the heck are we going to make that genetic material end up in both daughter cells? How are we not going to have 2/3 of it in one side and 1/3 in the other? That's not going to work. It's okay if we don't have a full mitochondria in both, one less in one side, we can just make another one. That's fine, but the genetics, we can't change that. And so we have this process, these phases that describe that process. And we have a prophase, a metaphase, an anaphase, and a telophase. So they name these things. They name the prophase, which means before, metaphase means in between, and anaphase means upward. I don't know where that comes from. And the telophase means an end goal. So there's some words, why they use those words. But so basically now what you have is you got a spaghetti soup of these chromosomes, and they become visible and get to like this, where we can actually see them in these chromosome type structures, and the nucleus also disappears, And that gets us then to the next phase, the metaphase, which is where the centrioles go to the corners and make these interesting spindles. These are like real freeway tracks. These are like mobile roads, you can think of that. So they reach in to an area in the center of this cell called the equator, equatorial plane in the middle, where chromosomes attach and line up by those centromeres where the legs cross. In a video where that is actually visualized, it's called kinetic core. So there's different names for this. But anyway, from there then, whenever things are ready, we're gonna have this anaphase, which is when the chromosomes have separate and they literally, you see it in a video, they get pulled to one side by molecules that pull them like on a rope and walk on these they call them asters, but I think of them like roads, basically, tracks. And then they go to each side of the cell, where the center, and then they split. And then the telophase is basically we make a nucleus again, and we sort of uncoil them and recreate the situation of them. So it's really basically the process of how do we split these chromosomes apart. So you go through those a few times. Sensible, okay, after. Does it make sense? I know after protein synthesis, that's no thing, right? Easy peasy. And then we also have to briefly talk about meiosis, and that's a reduction. What time is it? Oh, I gotta get moving. That's a reduction or maturation division. We use that when we make the sperm and the eggs. So how the heck we gonna get from 46 to 23? Because if you got a sperm and an egg come together, you can't have 46 in each side. You're gonna be way overdoing it. So you're gonna only come with 23 from each side, from mom and dad, and then combine and make 46. And so you gotta figure out how we're gonna get from the regular 46 down to 23. So basically what we're gonna do, more or less, is we're gonna have like two divisions. The first is like mitosis. We copy stuff and it gets distributed to two daughter cells. Except in this situation, some fragments cross over, which gives us diversity. That's why your siblings don't look the same. For sure, don't act the same. And then the second one is where you make a cell division without copying the DNA, and you end up with 23 because you split those in half, so that's 23. So that's basically what the meiosis is about. So mitosis is for all body cells. Meiosis is only for the gametes, only for the sex cells, the sperm and the eggs. And then we leave that alone. Because to finish up, we got to talk about transports. Substances need to move in and out of the cell. Yeah, that's very important. We have two types of transport. We have active transport, we have passive transport. Passive transport means it just happens. So that could mean concentration distributes. Things just dissolve. Like you put a sugar cube in water and you swirl it around, that stuff dissolves in the water. That's doesn't need any energy. It just happens. Active transport means it needs energy. ATP. Like if you would want to take your bike and come up the hill, you would have to really put a lot of energy out and to go up the hill. So very often we use this when we have to push molecules against a concentration, against a strong concentration that's already there. So it's like we have to push it into something that it naturally doesn't want to go to. And that's a matter of concentration. Have you ever been in the Tokyo subway? Probably not, right? They have pushers there. They fill these subways trains so much that before the doors close, they have these people that push them in before the doors can close. And so the concentration of people in that train is really heavy. So when those doors open, they all fall out. And so that's that concentration question. So if you like stuff a lot of sodium ions on one side of a cell membrane, and then all of a sudden you open a channel, everything wants to come in. They all want to go to one side where there's nothing there. So molecules want to move from a higher concentration to a lower concentration naturally. without using energy. And we make use of that in the body. So but that's the active and passive question. So are you going with the concentration gradient or against the concentration gradient? Is it using energy or not? And then we have a few different types. So we have passive transport, simple diffusion. That means the sugar molecules. You go from a higher concentration molecules naturally to a lower concentration. Why? Molecules want to be balanced out. They want to equal out. They want to disperse and intermingle. We kind of know that intuitively, but chemically we still have a concept with that. And then we have more diffusion, but this time it's not just free and simple and the sugary molecules. Now it's facilitated. So this is a place where you have, let's say, you have a cell membrane with a receptor, a protein channel in it that's going to let the glucose in or out. So when that protein channel is open, that glucose channel is open, it's going to go in and it's going to distribute according to concentration gradient. So if there's more sugar on the outside in the bloodstream and the doors are open, it's going to bring it all in because the concentration is high up here. And that's where the cell's even smart. The cell, that's where there's an exchange reaction. So glucose comes in, it tags it with a phosphate, and so then the concentration looks different because it's a glucose phosphate. That's a different molecule. So it always appears like when these are open, there's always more sugar in the bloodstream than inside of the cell. So it always comes in. So it's kind of smart the way the body does that. So those are passive. Once that channel is open, it just happens. You don't have to do no energy, nothing. We also then have a couple more passives. One is easy, one's filtration. That's like you're making coffee. So water and dissolved particles are pushed through a membrane pore system, separating particles from the solution. So this has to do with how thick are the pores in the filter that don't let the coffee grind go into the water, so do that. So it's not, what stays behind. We use that in the kidneys. The blood is filtered through a membrane like slits that only let certain things in and not all of it. That's filtration. But then we got osmosis, Jones. Osmosis is the diffusion of water between two compartments separated by a semi-permeable membrane, not letting solutes pass. In this situation, water will move to create an even concentration of the solutes in both compartments. That gets a little more complicated. So now what we have is we have a beaker with two parts, and we have this membrane here that does not let the particles go through. Only water can go through. The desire of this situation is that both sides have an equal distribution of the particles. That's just what nature wants. And so now, though, because we can't move the particles, it's smart. Now the water moves, not the particles. That's osmosis. We can use this in the body greatly. Like you have blood, the heart pumps the blood, and it goes into the blood vessels, it goes into the tissues, it leaks out. How's it going to get back into the bloodstream? How are we going to get it back into the veins? We let some molecules stay behind in the blood, then they suck the blood back in. So it's a suction. You can think of these particles become a suction of water thing. And you visualize that with, have you cut a tomato and put salt on top?
I don't like tomato.
Well, too bad for you. That's good for your prostate, brother. Anyway, lycopenes. No, but if you do that, you have first a little salt kernel, and all of a sudden it's a little drop of water on there, right? The salt pulls water towards it. So in the body, salt is brilliant to pull water out and towards it. And you see that in the tomato. So we have, for example, a situation where in the kidneys where we have sodium gets extracted from one side and water follows it. So we use that concept. So we use osmosis to have liquids move around in the body, and one of the main ones is water, of course. Boom. Now we get to the active one. So now we need energy. So we're going to go against the concentration gradient. So the biggest ones in there that we use all over in the body are these pumps. And these pumps, what they do is they actively carry small molecules against the concentration gradient that establish a concentration gradient for later use. So this is brilliant. So now what we do is, this is a nerve impulse, for example. So in a nerve impulse, we got, this is a cell membrane. So now we got these pumps, they're right, that's a pumping. The pump carries 3 sodiums to the outside and 2 potassiums to the inside. It's one of the pumps, the sodium potassium. We need those 2 ions to make a nerve impulse. And what happens is, with this always working, you create a high, heavy concentration. There's a big Na. Sodium is Na. On the outside of the cell, and very little on the inside. Now you want to make a nerve impulse. All you got to do is you got to make a gate and open the gate and let sodium come in. And the concentration gradient is so great that this sodium wants to come in all at once. It's so powerful. And what has sodium on it? It's got a charge. And that's how we make electricity move around in the body. So the electrical charge goes from all the way from the outside and gets sucked into the inside of the cell, and that moves, that's electricity moving. And so the electricity ends up being a passive event because the active pump establishes that high concentration gradient. So often, ATP-needing processes like a pump takes a lot more time than something that just happens naturally, like, you know, you're falling off, jumping down. Gravity pulls you down real fast. It's much harder to walk uphill before you can jump down again. Not the best example. Maybe we'll use the example of like you taking your bike up. Next Monday, we should all come to school with a bike. See how long it takes us to go uphill. Lots of ATP. But once we go down, it's like, make sure these wheels stay on, huh? It's like an explosion can happen. Nerve impulse is the same way. It's a really steep hill. We'll talk about that some more later. But this, I hope, is the last. Yes, thank the Lord. The cells, the more active processes are bigger stuff, like eating stuff. So when we use the word endocytosis, that means some substances get brought into the cell. The word endo means bringing things into the cell. The word exo is for exit. Exit is get out of here. So that leaves the cell. Exocytosis leaves the cell. So there's our substances. So when something like a food particle comes in, often what happens is, look at this, it's pretty, right? It's pretty. We have food particles coming into the cell, and the cell says, oh, look at that, we can bring this in. So it pinches off some cell membrane, and it has the food particles in cell membrane. Now what we need to do is we're going to get that lysosome, and we fuse the lysosome together and make a food vacuole lysosome combination. And then the food is being digested from the lysosome right inside of it. Or when we want to exit something out of the cell, like we made something and want to let it go. We talked about that with the Golgi apparatus. We just fuse this membrane with the cell membrane, and the contents can go out. Brilliant. Simple. When we eat, the cell eats something, that's known as phagocytosis. Cellular eating is phage is eating. PHA is that stuff is eating, phage. Pinocytosis means cellular drinking. And I remember that because there's Pinot Grigio and Pinot Noir. And I like those too. And so pinocytosis is drinking. So that, mnemonics got to be a little silly and then it works. Phagocytosis is eating, pinocytosis drinking. And that basically talks about this processing. Whew! Oh, that was a lot. How are you doing?
Good.
Hanging in there? Any questions come up right on top? No? Yes? Well, that's a no. That's good.